Hi everyone, my name is Prasanna from Andhra Pradesh, India. This is undergraduate inbound credit mobility program, integration of University Malaysia Kellington with Angro. My arrival to UMK. We reached to UMK Kottabaru campus. Welcoming ceremony was held on 14th Feb and next day 15th Feb we reached UMK Jelly campus. Training started on 16th Feb. This is the picture of UMK Kottabaru campus where welcoming ceremony was held and UMK Jelly campus. My personal information. My education. Fourth year BTech Food Technology in Acharya Engineeringa Agriculture University. My mentor Shaiful Azman, Field Supervisor Dr. Fatima Kayat. Location of training. This is the tissue culture laboratory and this is the nursery. My project is on plant tissue culture. What is tissue culture? It is a technique of growing cells, tissues, organs, or whole organism in vitro on artificial culture media under aseptic and controlled conditions. What is micropropagation? It is a practice of rapidly multiplying stock plant material to produce large number of frozen tissues using modern tissue culture methods, widely used for orchid ferns, etc. Steps of micropropagation: selection and preparation of explants, initiation of culture, multiplication, rooting, and plant development. List of activities: MS media preparation, orchid subculture, orchid nursery management, growing, ginger sowing. Water orchids. The orchid family is one of the largest groups of flowering plants on the planet, with around 30,000 species growing in the wild and well over 120,000 man made hybrids. There is no shortage of choice in the orchid world. Orchids are able to live about 20 years depending on the type of orchid and the environment. I was assigned to Cymbidium orchid, which is a kingdom Plantae, phylum Tracheobionta, class Liliopsida, or order Orchidales, family Arachidaceae, genus Cymbidium. Cymbidium. Cymbidium also called boat orchid. Genus of 50 to 70 species of tropical and subtropical orchids. The genus is primarily distributed in Asia, though several species are native to Northern America. Cymbidium species are evergreen and grow in soil or epiphytically on other plants. Most have short pseudobulbs, bulb-like stem bases, which is storage organs for minerals, water, carbohydrates. Stripe-like leathery leaves and many flowers on each flower stalk. The plants are known for their showy flowers, which range from white, pale yellow to green, maroon, and dark bronze, and can persist for several months. The cut flowers are also fairly long lasting and are commonly used in cosmetics. Media preparation. The success of the plant tissue culture experiment largely depends on the selection of right culture media. To induce organogenesis and regeneration of plants, Mushragan's cook is widely used. The pH should be maintained at 5.8. If pH is less than 5, ability of explant to take up nutrients will be affected. If the pH is greater than 7, iron will out of media, becoming unavailable for uptake. So pH should be maintained at 5.8. The preparation of half strength MS media. Here is the materials and three types of media: multiplication media, elongation media, and rooting media. In multiplication media, PGR is used, which is about 0.5 mg per ml BAP. And in rooting media, activated charcoal is added, which is about 2 grams. Different medias. These are the pictures of different medias: multiplication media, elongation media, and rooting media. Subculture of Cymbidium. Subculture is the removal of media and transfer of explants from the previous culture into fresh media to prolong the life and growth. Subculture is done in laminar airflow under sterile condition. The whole procedure done under aseptic condition to avoid contamination. Subculture of Cymbidium. This is the explant placed in media. Acclimatization. Acclimatization is the adaptation of plantlets to a new environment. Plantlets or shoots in the culture vessels are customized to a different microenvironment. These are the process steps. Firstly, the in vitro rooted plantlets were removed from the culture and washed thoroughly with running tap water to remove the attached media and place them in a tissues placed box. Then transfer the explants to the planting material containing charcoal, coconut chunks, and then transferred to the nursery. If the plant is affected or infected, which were placed in the therum, which is a fungicide, for 30 minutes and placed in your tissues, then spray water on it, and which is observed for 2 to 3 weeks. Then transfer the explants to the planting material containing pots and then placed in the nursery. This is the picture of cymbidium plantlets and preparation of planting material in nursery and acclimatization of plants. What is housekeeping of nursery? The sum of the activities performed for the successful production, care, and marketing of different planting materials like seeds, seedlings, cuttings, etc. Nursery activities are weeding, watering, in vivo propagation, preparation of planting material, arranging of pots on beds, ginger sowing. This is the picture of in vivo propagation which we done in nursery and housekeeping of nursery. These are the pictures of ginger sowing. My learning outcomes are in detail about plant tissue culture and knowing about subculturing of orchids and different types of subculturing of different orchid plants like cymbidium, oncidium, dendrobium, and preparation of Mostragan Skoog media preparations, which is full strength media or half strength media, and nursery housekeeping. Thank you.